From the moment of conception, the human body orchestrates a microscopic marvel, the rapid replication and differentiation of cells to build an entirely new organism. But this process presents an immediate biological conflict. Because the fetus contains foreign DNA from the father, the maternal immune system would normally recognize it as an invader and attack. To prevent this rejection, the mother's body deliberately downregulates its own cellular defenses. This chart tracks maternal T-cell immune response against fetal tissue acceptance over the 3 trimesters. To ensure fetal survival, the mother's immune defenses drop significantly, creating a persistent vulnerability that lowers the threshold for standard foodborne pathogens to cause systemic damage. The placenta operates as a formidable, highly selective biological checkpoint, filtering maternal blood before it reaches fetal circulation. Yet, despite its structural density, the placenta remains highly permeable to specific microbiological and chemical agents equipped to bypass this barrier. We will analyze 5 specific dietary vectors, compounds and organisms that are completely benign to a healthy adult, but possess the exact biological mechanics necessary to breach the maternal-fetal boundary. The danger emerges specifically from how these microscopic loads interact with an altered, highly permissive maternal environment. The first vector is bacterial transplacental infection, primarily sourced from unpasteurized dairy, such as raw milk and soft cheeses. These products can harbor Listeria monocytogenes. Unlike most bacteria, Listeria thrives at standard refrigeration temperatures, and its resilient cellular walls allow it to bypass normal maternal gut defenses. This schematic illustrates the molecular breach. Listeria utilizes specialized surface proteins, internalins, to bind directly to placental host cell receptors. This triggers the host cell to engulf the bacteria, initiating a cellular breach and transporting the pathogen directly into the sterile fetal environment. Inside, the fetus has no developed immune system to stop the replication. The infection targets immunologically naive tissue, leading to severe complications, blood infections, or premature delivery. Pasteurization serves metals, ingested via bioaccumulation in high-level predatory fish, such as shark and swordfish. Methylmercury is particularly dangerous because it is lipophilic. While water-soluble toxins are easily filtered and excreted by the maternal kidneys, fat-soluble compounds persist and accumulate in the body's tissues. Because cell walls are composed of lipids, methylmercury diffuses effortlessly through both the lipid-rich placental membrane and the developing fetal blood-brain barrier. Once inside the brain, the mercury molecules physically block the assembly of cellular microtubules, the structural scaffolding that neurons require to form and connect. Without these microtubules, cells cannot migrate or divide properly, leading to permanently arrested brain development. Consuming apex predators passes concentrated environmental toxins directly to the most vulnerable neural architecture in existence. The third vector is systemic maternal compromise, frequently triggered by Salmonella bacteria found in raw or undercooked eggs. Unlike Listeria, Salmonella rarely breaches the placental boundary to infect the fetus directly. The threat stems from the maternal body's reaction to the infection. Salmonella induces a severe systemic response involving extreme hyperthermia and intravascular dehydration. This fluid loss triggers a physiological cascade. Dropping maternal blood volume forcefully restricts placental perfusion, narrowing vessels and slowing blood flow to the womb. This restricted flow deprives the fetus of oxygen, inducing acute fetal hypoxia and significantly elevating the risk of immediate miscarriage. A severe threat to the mother's vascular stability constitutes an immediate, life-threatening crisis for fetal viability. The fourth vector is parasitic teratogenesis, transmitted via the Toxoplasma gondii protozoan found in undercooked meat. The transmission is stealthy. Once the maternal digestive tract breaks down the infected meat, surviving parasites enter the bloodstream and cross the placenta into fetal circulation. Inside the fetus, the parasite embeds deep into soft tissues, actively multiplying and forming permanent, hardened cysts. In the brain, these cysts trigger localized cerebral calcification, replacing healthy brain tissue with rigid, calcium-dense deposits. A secondary damage pathway deeply affects ocular development, where parasitic cysts lead to retinochoroiditis, driving severe tissue inflammation and permanent scarring. Failing to thermally destroy parasites in muscle tissue prior to consumption maps directly to permanent sensory and cognitive deficits in the child. The final vector is vasoactive chemical overload, driven by excessive maternal caffeine consumption. Caffeine molecules are small enough to diffuse freely and rapidly across the placenta. This chart highlights a major processing disparity. The fetus entirely lacks cytochrome P450, the specific enzyme required to metabolize caffeine, leading to an exponentially rising accumulation of the chemical in fetal circulation. This accumulation causes continuous overstimulation, resulting in a chronically elevated fetal heart rate. Caffeine also acts as a vasoconstrictor, tightening the blood vessels within the placenta and restricting the flow of nutrients required for normal fetal growth. Mild maternal stimulants become profound stressors when the biological tools to break them down do not exist. Mitigating these 5 vectors requires shifting from awareness to strict clinical action, framing dietary choices as the implementation of an external biological filtration system. Subjecting meat and eggs to high temperatures ensures thermal destruction of bacterial and parasitic vectors, neutralizing toxoplasma and salmonella before entering the maternal system. Chemical pasteurization remains the only reliable molecular filter, capable of halting the advance of adaptive pathogens like Listeria. Managing neurotoxicity requires active auditing of seafood intake, limiting consumption to species with verified low mercury profiles. For vasoactive chemicals, strict pharmacological dosing is required. Capping caffeine intake at 1 cup per day prevents toxic accumulation in an enzyme-deficient fetus. These dietary restrictions are the foundational architecture for maintaining the integrity of the maternal-fetal boundary. Systemic symptoms during gestation, specifically a sudden fever or acute nausea, severe gastrointestinal distress, are critical medical alerts. These symptoms mandate immediate medical consultation and intervention to prevent the rapid escalation of placental risks. Maintaining strict biological and nutritional boundaries dictates the success of early development. Informed dietary decisions ensure maternal fetal security and the delivery of a healthy life into the world.